The next session in our symposium focuses on novel methods for improving our ability to accurately identify health outcomes in safety surveillance activities. This work leverages the existing Sentinel system, but has broader applications to safety surveillance activities. In this session, we will be hearing from Dr. David Carrell and Dr. Susan Gruber. Dr. Carrell is an associate investigator at Kaiser Permanente Washington Health Research Institute. His research uses clinical natural language processing methods to extract information from unstructured clinical text to support precision phenotyping, the automated identification of health conditions and outcomes of interest through the extraction and analysis of rich information available in electronic health records. Through collaborations with researchers bringing complementary expertise in clinical, informatics, and statistical domains, he has applied precision phenotyping methods in the areas of cardiovascular disease, cancer and cancer risk factors, substance use disorders, mental health conditions, and medical product safety surveillance. Dr. Gruber is a biostatistician, computer scientist, and founder of Putnam Data Sciences, a statistical consulting firm specializing in causal inference and predictive modeling. Her work focuses on the development and application of data adaptive methodologies for improving the quality of evidence generated by observational healthcare studies. Dr. Gruber is the former director of the Biostatistics Center and assistant professor in the Department of Population Medicine at Harvard Medical School and Harvard Pilgrim Healthcare Institute. Prior to that, she served as senior director of IMED's methods research at the Reagan-Udall Foundation for the FDA. Welcome, Drs. Carrell and Gruber.
Hi, we're happy to be presenting to you today on our topic, electronic health records plus natural language processing and machine learning equals improved Sentinel outcome detection algorithms. I'm David Carrell from Kaiser Permanente, Washington, and I'm presenting today with my colleague, Susan Gruber. To give you an overview of what we'll be talking about today, first we will present the motivation and goals of this work as a project to develop models for detecting anaphylaxis. We'll talk about the study design and electronic health record data. I'll go into a fair amount of detail about natural language processing of clinical notes, and then Susan will present to you the machine learning models for outcome identification. So first, the motivation and project goals. Some of you may already be familiar with ARIA, the FDA's active risk identification and analysis system designed for post-market safety surveillance. ARIA has two main features: a set of predefined, parameterized, reusable analytic tools used to query data and conduct analyses of that data. And secondly, electronic claims data and its equivalent extracted from electronic health records, structured according to a well-defined common data model. This is the Sentinel common data model. These two features allow Sentinel to quickly and easily execute distributed programs across a network incorporating data on more than 100 million lives to perform risk identification analyses. You may also know how ARIA is used. When there's a serious safety concern that falls under FDA authority and observational data are considered appropriate, the FDA considers whether the ARIA system is capable of assessing that safety concern. If ARIA is deemed sufficient, that's the upper path on this slide, then a safety analysis is conducted in Sentinel using ARIA. If ARIA is considered insufficient, the lower two paths, the FDA can issue a PMR, or post-marketing requirement, that the sponsor, often a pharmaceutical company, conduct a post-marketing safety study. And another option is for the sponsor to conduct something called a related ARIA study. In any case, a lot depends on the question of whether the ARIA system is sufficient. There are several reasons why ARIA may not be sufficient. An FDA-led evaluation of over 200 safety issues, about half identified before the product received FDA approval and half after, found several reasons for ARIA insufficiency. which are summarized in these boxes. Inability to accurately identify the relevant study population was a common reason. Inability to measure important exposures or covariates were also common. Sometimes the study required analyses the ARIA tools were not designed to perform. The most common challenge, appearing in about 70% of issues where ARIA was not sufficient, was the inability to accurately capture the outcome, the health outcome of interest, using claims data. Anaphylaxis is one such health outcome of interest for which ARIA is insufficient, and that's why we're studying it. Robert Ball and others at the FDA have studied ways to improve capturing anaphylaxis outcomes by augmenting claims data with information extracted from electronic health records using natural language processing. However, so far, these approaches have yielded similar PPVs, or precision, around 60%, as earlier approaches using only claims data. But by examining the cases where automated algorithms were making mistakes, this work also suggested possible approaches to improve the capture of anaphylaxis, as noted here on the right. They include things like capturing information about the timing of symptom onset, or information about the severity of symptoms, information about the presence of alternative explanations are also important. These findings motivated our study to better measure using NLP and better interpret using machine learning these types of information. We have two goals. Our primary goal is to improve classification accuracy of a health outcome of interest to Sentinel, which is anaphylaxis. As we've already noted, this will be challenging because anaphylaxis is extremely rare, occurring in about 36 per 100,000 person years. It's a complex diagnosis, which can be difficult even clinically to recognize and accurately diagnose based on a complex symptomatology. And it's a condition that resembles other conditions, such as severe allergic reaction, which is not anaphylaxis. Further, anaphylaxis is often not coded accurately. Only about 2/3 of diagnostic codes and claims data are for true anaphylaxis cases. Our second goal is to use this work to inform how Sentinel can improve classification of other health outcomes of interest to Sentinel through methods and reusable tools. What are the key areas of methodological innovation we will use? The first is leveraging more data from electronic health records. As you all know, the electronic health record is a richer and much more nuanced source of information than structured medical claims. And the widespread adoption of EHRs, most of which has occurred after Sentinel was originally conceived, makes this possible. The second is applying NLP methods to unstructured electronic health record text or clinical notes, which will hopefully allow us to extract information needed to improve classification of outcomes of interest. The third innovation in this work is using machine learning, which allows more automated, more flexible, and data-driven modeling approaches. Now let's briefly look at the design of this study. During the October 2015 through December 2018 period, which is all within the ICD-10 era, we conducted this study at two study sites, Kaiser Permanente, Washington, and Kaiser Permanente Northwest. We sampled from a population of adults and children. We obtained gold standard outcomes via manual chart review, and we specified and operationalized various structured data covariates using the existing Sentinel Common Data Model. And additionally, we extracted NLP-derived covariates from free text clinical notes. We used stratified sampling to create our study sample. The big dark blue circle here represents all visits at the two sites during the study period. The lighter blue circle is for eligible visits, which includes some true cases marked here with stars, but mostly non-cases. Now, anaphylaxis is rare, so instead of randomly sampling from this entire light blue circle, We predefined a stratum, the lightest blue circle, where we expected more cases to be, and we oversampled from this high information stratum, or path, as we called them. We originally thought we would have 3 paths. Path 1 required an inpatient or emergency department visit with a diagnosis code for anaphylaxis. Path 2 required the same diagnosis, but in an outpatient visit. And path 3 included inpatient or ED visits that didn't have an anaphylaxis diagnosis, but did have other diagnoses we thought might be concealing some true anaphylaxis cases. These were diagnoses for severe allergic reactions or adverse drug events. It turned out that path 3 yielded only 1 true case out of about 100 charts we manually reviewed. So we didn't include it in the work we're presenting today. What we did do is we created an analytic data set by randomly sampling from path 1 and randomly sampling from path 2 to create our data set, which we refer to as the analytic data set. Now I'd like to dive a little deeper into the natural language processing methods that we used. We define clinical NLP as an approach that's guided by clinical domain knowledge, combines methods from computer science, artificial intelligence, and computational linguistics to derive information from unstructured clinical text and represent that information as structured features. Those features can then be used to develop automated algorithms. In our case, these NLP algorithms identified things like exposures, covariates, and other evidence we hoped would be useful for distinguishing true anaphylaxis cases from non-cases. Here we can see the major steps in implementing clinical NLP. The first step is assembling a corpus, which is the set of clinical notes that will eventually be processed by the NLP system. In our study, these came from an EHR system, but some of them came from paper copies of charts that had to be converted using optical character recognition. The second step is to create gold standard labels, answering the question, which visits are true anaphylaxis and which are not. And this requires manual review. The third step shown here with the dashed red line around it is to engineer NLP features and algorithms. This is usually an iterative process that involves trying out, evaluating, and modifying the various pieces that go into an NLP system. The dictionaries, the logic, ways to identify negation, and various other things. Feature engineering, which we'll talk about a lot in this presentation, is the process of determining which textual content will be converted to structured NLP features or covariates. In this study, we engineered NLP features manually based on clinical expertise and informatics expertise and knowledge. The goal of that work was to identify content that is both clinically relevant to the outcome of interest and also feasible using the NLP technologies and the data that are available. There's a broad continuum of approaches that can be used to develop NLP systems, from manual to semi-automated to fully automated. We won't go into the automated approaches today, but it's important to be aware of them. In a separate project that Susan and I are involved in, we are investigating whether some of these automated NLP approaches may be useful in Sentinel. As I mentioned, in this study, we had to convert a very large number of paper charts to electronic text using optical character recognition, or OCR. I won't dwell on the OCR component today, but OCR is something that should be done only if it's necessary. And because OCR isn't perfect, it can introduce errors into NLP data. As we can see on this slide, errors include things like misinterpreting characters, which can completely disguise an actual word. Here we see an example where bowel became Bowie with an exclamation point, and that exclamation point terminated a sentence that otherwise would have continued. Formatting problems are also common in OCR. In this example, a key phrase, severe anaphylaxis, is split across two physical paper pages. The page header and footer text makes it nearly impossible to accurately capture this statement. In this study, we used two different approaches to rate our gold standard data. At Kaiser Washington, we used dual blind review by clinicians, and their decisions were recorded simply in Excel spreadsheets. At Kaiser Northwest, we used a more sophisticated approach that we thought might scale better to other Sentinel projects. We used dual blind review by non-clinician abstractors who followed a protocol. Abstractors decided the easy cases, which was about 90% of all the cases requiring abstraction, and then clinicians adjudicated the more difficult cases. We also used REDCap forms at Kaiser Northwest to record the decisions and, importantly, to preserve the supporting documentation for those decisions, which might be useful in future work. As we saw a couple of slides ago, NLP systems use dictionaries to identify important content. And we use several complementary approaches to create the NLP dictionary used in this project. We use manual review of charts by clinicians and informaticists. We use something we call NLP-assisted query of the clinical corpus. We got synonyms from other medical dictionaries. And we also used a semi-automated approach that leverages information in clinical knowledge sources. I'll describe each of these. For manual chart review, our clinicians and informaticists reviewed, discussed, and marked up charts according to a think aloud protocol. We applied this to about 50 charts. And you can see here in these two pictures examples of markings that were made by one or the other clinicians or informaticists. In the second picture, you can see that often when anaphylaxis is present, the clinician will document several qualifying symptoms in a short span of text in the clinical note. Because we noticed this in several charts, we created NLP features for it. For example, we created a binary flag indicating whenever there were at least 3 distinct anaphylaxis relevant symptoms mentioned within a span of text that was no more than 200 characters long. That was an attempt to capture something like we're seeing in this second picture. Additionally, as I mentioned before, the dictionary terms that were focused on through manual review were those that were both clinically important and feasible for NLP. The second approach to dictionary creation was NLP-assisted query. NLP-assisted query is simply putting a corpus of text into a relational database that has full text indexing, such as Microsoft SQL Server or many of the other databases, and using that full text indexing to conduct queries and return results, as is shown in the bottom of the screen here. For example, we knew that dyspnea was an important concept, but we were curious how it might be used in real notes. So we started with some known terms like breath in shortness of breath and the same string of characters in trouble breathing, and identified notes in the corpus that had those character strings in them. We then deduplicated the results and reviewed them. And as you can see here, there were 208 unique strings that were returned by this query. And it included 5 new terms that were synonyms for dyspnea: cannot breathe, breathing problems, hard to breathe, couldn't breathe, stopped breathing. The third approach to developing our dictionary was to look up synonyms of terms we had already found in the Unified Medical Language System Metathesaurus. So as you can see here, the concept of dyspnea has many different synonyms: breathing difficulties, the abbreviation DIB, difficulty in breathing, and so on. The fourth approach was automated feature extraction from clinical knowledge sources. This is an approach that you and colleagues described in a 2015 JAMIA paper and a method they referred to as automated feature extraction for phenotyping, or AFEP. The basic assumption of this approach is that if you look in a collection of relevant knowledge-based articles about a specific clinical condition, the important terms will appear in a majority of those knowledge-based articles. So let's see how this is implemented. It starts with a collection of knowledge-based articles. Here are from the Mayo Clinic, Medline Plus, Medscape, and so forth, 6 different knowledge-based articles about anaphylaxis. The text of those articles is then processed through an NLP system that uses the SNOMED dictionary of clinical terms to identify all of the unique clinical terms that appear anywhere in any of those notes. As you can see here on the right, there were 367 unique SNOMED terms found in this collection of knowledge-based articles. 90 of those terms appeared in at least 3 of the sources. And so according to the logic of the AFEP method, those would be the terms that would be added to our dictionary. This is a summary of those terms. And all of these terms appeared in at least 3 of the articles. Some appeared in 4 and some in 5 or 6, as you can see in the left and middle columns. Highlighted in yellow and red is the concept we started with, anaphylaxis, the topic of each article. In light blue are some terms that are in the UMLS dictionary and also appeared in knowledge-based articles, but are not useful for NLP development because they're non-specific to anaphylaxis. Things like diagnosis, cells, disease, complications. Lists like this one generated by AFEP are easily manually reviewed to eliminate terms that are obviously non-useful or non-specific. Now, combining the terms we discovered through all 4 approaches gave us a dictionary with 843 distinct terms, the first 35 of which are shown here alphabetically. Interestingly, about half of these terms related to skin or mucosal involvement, a critical symptom in diagnosing anaphylaxis. Each chart, it turned out, had almost 130 instances of these terms. For example, if a chart had 3 mentions of the term wheeze, that would contribute 3 instances to its total. One chart had more than 2,000 instances. The concepts listed in the far right column of the table were used to form groups of terms that referred to the same thing and helped us engineer features. Since we were manually creating the features and covariates to be used in model development, we used our knowledge to develop features we believed would help distinguish true cases from non-cases. We identified all occurrences of each dictionary term in the entire study corpus. This yielded a huge amount of very granular data about each term, where it appeared in the text, whether it was negated, and so forth. From this granular data, we manually engineered our NLP features. For example, since we knew that it was important to capture when a chart described diagnostic criteria, we manually engineered some features to represent when this happened. This table summarizes the diagnostic criteria published by Sampson and colleagues for identifying anaphylaxis. There are 3 criteria. You can see here on the first criterion circled, the chart must describe either a combination of skin and mucosal involvement plus respiratory compromise, or skin and mucosal involvement plus reduced blood pressure. This therefore yielded 2 NLP features, as we can see there on the right. We also identified a number of other features. There were 72 relevant clinical concepts ranging from abdominal pain to wheeze alphabetically that we operationalized. We also operationalized some special features, things like a statement that a patient was admitted to the hospital for observation, or that the chart contained an explicit statement that the clinician believed the patient had anaphylaxis as a formal diagnosis. We also captured things about treatment, such as epinephrine, because those are important in treating the condition of interest. One of our rules in engagement was to not use any of the gold standard data to improve engineering of the features. We did this because we had a modest sample size and we wanted to reserve all of the gold standard data for modeling. Here we illustrate some of the rules that we used in engineering NLP features manually. Sometimes we used a rule of two, meaning if there were at least 2 mentions of anaphylaxis, then a binary flag would be set to true for that chart. Sometimes we use counts of terms, such as the number of mentions of airway restrictions, or the counts of all mentions, sometimes the counts of only the affirmative mentions. Sometimes we normalize the counts by the total amount of text that was available in a given chart, assuming that charts that were longer would have higher counts than charts that were shorter. We also created binary flags for mentions of individual concepts, as we saw in the dictionaries that I showed earlier, and binary flags for anaphylaxis symptom groups. For example, reduced blood pressure, gastrointestinal symptoms, respiratory compromise, skin or mucosal involvement, and a category for other types of concepts. As I illustrated earlier, some of the combinations satisfied diagnostic criteria, and sometimes we required the concepts to appear in a short span of text. These are the subgroups of NLP-derived concepts that we operationalized, and we created binary flags for 66 of them. The remaining 5 concepts were used to engineer something we called special features, such as whether a statement of an explicit diagnosis of anaphylaxis appeared in a chart, or whether the patient was admitted for observation. Other special features are described here. In addition to explicit diagnoses, we had special features for rapid decline, such as the description that the patient was collapsing or getting worse. We also had features for when a term conveying suddenness appeared near a term describing a symptom, such as rapid swelling. Other features captured need for observation and having an epinephrine prescription. Overall, we used 116 features for model development. These came from a larger set of 471 features that we had engineered, many of which we expect to be of low value. From these 471, we selected 100 that were considered to be the best features for capturing the content of interest. Additionally, we had our clinicians identify a set of 25 features they judged to be important. And that included 16 that were not in the top 100 identified by the informaticist. So overall, there were 116 features that were included in our analytic data set. As you can see here in the table, 30 of them operationalized diagnostic criteria. 66 were related to various anaphylaxis-related concepts. 10 were devoted to the critical organ systems involved in diagnosing anaphylaxis. 5 were for anaphylaxis terms themselves, including explicit diagnoses. And then 5 were for the special features that I described earlier. I've been talking a lot about the NLP features, but of course we also engineered structured data features. There were 47 structured data features, and these were also manually created jointly by the clinicians and informaticists. The categories of structured features are summarized here. Some had to do with high-risk exposures, such as exposure to imaging dye or immunotherapy. Several were for competing diagnoses, asthma, COPD, other conditions. Some were for treatments, either medications or procedures such as CPR. We had a couple of features for follow-up with immunology or allergists within 45 days following the encounter, because we believe this might be more common for patients who had true anaphylaxis. We also had features for the type of anaphylaxis, food-related, venom, and other. We looked back historically into the record to develop features for patients who had a history of anaphylaxis and also a history of allergic reaction. We captured whether the encounter was in the emergency department, an inpatient or an outpatient setting, and we operationalized demographic features. After operationalizing all of the structured and unstructured features, we combined them into an analytic data set. We packaged a very large amount of SAS code for the structured features and Python and SAS code to capture and summarize the NLP-derived features. We put this package and documentation up on GitHub and shared it with our collaborators at Kaiser Northwest so that this exact same data generated at Kaiser Washington based on manual creation of features at Kaiser Washington could be generated at Kaiser Northwest. That concludes the presentation of the NLP work. I'm now going to turn this over to Susan, who will describe for you the machine learning methods that we used in this study.
Thank you, David. So now you've heard about all the work that went into creating the analytic data set, and it now gets passed off to me to construct a risk prediction model to identify subjects who experienced the outcome of anaphylaxis. This schematic diagram shows the approach we took. In step one, we created the data set, and it consists of approximately 240 observations. Only 85 of these are non-cases. We also have nearly 140 candidate predictors. So as you probably already know, when there are more predictors than outcome events, a parametric mean terms logistic regression model will overfit the data and end up perfectly predicting the outcome for every subject in the data set. Of course, we'd like to avoid this. And one way to do that, to create a model that's based on true associations in the data rather than on random noise, is to carry out some sort of covariate selection procedure. Another way is to use a machine learning approach that incorporates some internal form of regularization. And we actually are going to try both of those routes. We're also interested in understanding the added benefit of incorporating the NLP covariates. And so we're going to compare models that are built based on structured data alone with those that are constructed using the structured data along with the NLP EHR covariates. So when we start with only 43 structured covariates, as is shown right here on the slide, that lessens our worries about overfitting, but it doesn't completely remove them because, again, we only have 85 non-events, 85 occurrences of the minority class. So step 2 in this diagram is a preliminary screening step that removes one of a pair of covariates that have identical values for each subject. For example, in our data, we have a binary indicator of same-day epinephrine administration and also a binary indicator of same-day vasopressor administration. These two variables had the exact same patterns of zeros and ones, and so one was removed from the data set. We also removed covariates for which 99% or more of the observations had the exact same value. Since we don't want to remove a covariate that's highly predictive of the outcome, we also required that the absolute correlation with the outcome was less than 0.05. And in this step, we ruled out about 4 or 5 variables. In step 3, we developed a model using superlearning, which is represented by the black box with the number 1 on it. And we'll talk about what's inside the box on the next slide. But I want to continue with the process. So after we fit the model to data, we can use it to obtain risk predictions on novel data. You can see in step 4 that the predictive risk for observation 1 was 0.92, so we labeled it a case. And then for subject 2, the predictive risk was 0.01, which we labeled as a control, and so on for all N observations. We'll talk later about how we chose the threshold for labeling someone as a case or a non-case. Now we want to compare the performance of that model with one that was fit to structured data and also the EHR text-based NLP variables. We'll follow the exact same procedure. And then in step 4, we get out novel predictions. They're not the same as from model 1 because they're based on different predictors. You can see that for observations 1 and 2, the predicted probabilities from the 2 models are in close agreement, and the class labels are identical. But now look at observation 3. Using model 1, its predicted risk of 0.84 was high enough for it to be labeled a case. but the risk of 0.63 from model 2 caused it to be labeled a control. We're not surprised that there are some different predictions and different classifications from the two models. The predicted risk scores are somewhat correlated, but obviously they're not the same. So which model produced the better set, the better risk scores, is something we'd really like to know. But before we look at those results, let's talk about our superlearning approach to constructing the models in the first place. As this slide shows, inside the black box are different base algorithms and also superlearner, which is a weighted combination of them all. We tried variants of logistic regression and penalized logistic regression. It's not a good idea to use only a single pre-specified logistic regression model because it may omit important interaction terms or meaningful transformations of continuous variables. However, we also don't want to exclude them from consideration either, because if it happens to be a good model, then its familiar form is a bonus in terms of interpretability and acceptance by clinicians and other healthcare professionals who might be using the model in the future. You may also have heard of lasso and ridge regression. Elastic net is a generalization of those approaches that also produces a logistic regression model. But this is penalized fit, and so the coefficients in the model are shrunk towards 0, which helps reduce the potential overfitting to data. We also see two tree-based algorithms, Bayesian additive regression trees and boosted trees. These are non-parametric approaches that have internal regularization procedures to also avoid overfits. And finally, we tried neural network. Neural networks typically require a lot of data because there are many weights to be learned internally. So we didn't have high hopes for success with neural networks, but we did want to see what would happen. And again, the last approach was the ensemble superlearner, which is a machine learning algorithm that uses cross-validation to calculate an optimal weighted combination of predictions from the other algorithms under consideration. And those algorithms taken together are also called the superlearner library. Some of the algorithms require setting what are known as tuning parameters, and they give different results depending on the values of these tuning parameters. Column 1 of this table lists the 6 different types of algorithms we considered, and then column 2 shows the R package name. And in column 3, we list the important tuning parameters settings for variants of the same algorithm. So our superlearner library contained 8 variants of these 6 base algorithms in all. At the bottom of the slide how we built 75 different models for predicting anaphylaxis. And ultimately, we choose the best model based on a comparison of the cross-validated performance. We considered 3 separate data sets. You already heard about the first 2 of these. The first one contains only the structured covariates. The second one contains the structured covariates plus 116 NLP-derived covariates from the EHR. We also considered a third data set that had the structured covariates and a subset of the NLP-derived covariates that were selected by clinicians. We allowed them to select up to 25 covariates. Clinical domain knowledge played a role all along. The physicians were key in deciding what data to consider from all the information available in the claims and EHR data. But we also know that physicians often have good insight into the important covariate outcome relationships that are predictive. And so we wanted to compare the predictive information in this data set with the 25 NLP-derived covariates selected by the clinicians with the other 2. Because dimension reduction is important, we coupled each algorithm with 3 different covariate selection or covariate retention strategies. The word none here is equivalent to a strategy where we had no covariate reduction, or think of it as retaining all the covariates. The lasso screening approach retained only those covariates that had non-zero coefficients in a lasso model. So these are associated with the outcome, and it also takes into account correlations among the covariates so that 2 that are very highly correlated, only one will end up in the model and the other one will be excluded. The selection strategy was based on partitioning around medoids, or PAM clustering. With this approach, you cluster the variables into different groups and select one representative variable from each group to retain in the data set. We considered creating between 3 and 20 clusters and use what is known as a silhouette width to pick the best number of clusters to create. The silhouette width is a summary measure that balances how cohesive each cluster is with how far apart the clusters are from one another. Altogether, this gives us 24 base learners, coupling the 8 in the library with the 3 retention strategies, plus 1 super learner, that's 25, for each of 3 data sets, producing 75 candidate models in all. Once we have the models, we use cross-validation to compare them and pick the best one. When you're developing a prediction algorithm, then this is going to be true whether you use machine learning or whether you just use a parametric approach. If you train the model on n observations and then assess empirical performance on those same n observations, you can easily obtain an overly optimistic assessment. In this case, if you applied that model that looked very good to a separate data set, even one that was drawn from the exact same superpopulation, you would see much more misclassification than you expected. And that's because you based it on empirical performance. The solution is to focus on cross-validated performance. So you need to evaluate how well the model performs on a validation set that is independent of the training set. There are many forms of cross-validation, and we used v-fold cross-validation with v set to 15. And this is a very efficient use of the data, and it is probably preferred over creating just a single hold-down data set, particularly when we only have 240 observations. So with 15-fold cross-validation, what this means is we divide the data into 15 disjoint sets of equal size. We train on 14 15ths of the data, and then we assess how well the model performs on the held out 1/15th of the data. But we don't just do that once. We repeat that 14 more times, each time evaluating on a different held out 1/15th of the data. We find the average performance across all 15 independent validation sets, and that will be our cross-validated metric. What we focused on was a global performance measure known as the AUC, the area under the receiver operating curve. AUC measures the ability to properly distinguish between true cases and non-cases. An AUC of 1 is perfect discrimination. An AUC of 0.5 is no better than chance. It's like a coin flip. So most models will be between 0.5 and 1, with 1 being the most desirable. We also evaluated performance on independent data from a second healthcare facility, Kaiser Northwest. You can see the number and proportion of cases and controls at Kaiser Washington on the left. This is where the data we constructed the model were collected. On the right, you see the same information for Kaiser Northwest. Though not identical, the distribution of cases and non-cases in each path and overall is similar. This plot shows the v-fold cross-validated AUC for several of the models, along with their 95% confidence intervals. If we start at the bottom in pink, we see that the best model constructed using the structured data alone was constructed with a neural network coupled with a lasso pre-screening algorithm. The cross-validated AUC was 0.62. Remember that 0.5 is no better than chance, so 0.62 is moderately successful. Now moving up, if we allow the algorithms to consider the 25 clinician-selected NLP covariates along with the structured data, this improved the AUC by nearly 10%, as shown in yellow. The best model constructed using this data set was built by BART, coupled with a lasso pre-screener. Then the top 2 show the results when we allow the algorithms to consider all 116 NLP-derived covariates along with the structured data. The AUCs improved even further to 0.70 as shown in blue and 0.71 as shown in green. Both of these are BART models coupled with different pre-screening strategies. The lasso pre-screener was slightly better on the KP Washington data, but the confidence intervals are nearly identical. They have nearly complete overlap. And so in a sense, these models are fairly indistinguishable. The lasso pre-screener retained only 40 covariates. So for that reason, we might prefer it, since in practice, it's a little easier to only have to assemble 40 variables instead of 137 that are required for the retain all strategy. However, we also looked at model performance on the KP Northwest data. And when we did that, we saw that the BART2 retain all model generalized better, with an AUC of 0.67 when evaluated on the Kaiser Northwest data. Although I don't have it on the slide, I believe the AUC of the BART2 Lasso model was only 0.64. Thus, when we consider cross-validated performance at both sites, that leads us to choose the BART2 retain all model. The next step is to choose a prediction risk threshold for classification. If risk is greater than or equal to this probability, then we'll classify as a case, otherwise as a non-case. For purposes of an FDA safety study, we are most interested in a positive predicted value, or PPV, of 80% or greater. PPV is the probability of truly being a case if classified as a case. It's also desirable to have high sensitivity. In other words, to identify a high proportion of the true cases. This plot shows 4 cross-validated metrics evaluated on the KP Washington data. Along the x-axis is the percentile of the predicted risk score. The y-axis is the value of the metric between 0 and 100%. PPV is shown in red, sensitivity is shown in yellow, and 2 additional metrics are the specificity shown in blue and the negative predictive value shown in green. Specificity is an analog of sensitivity. It's the proportion of true non-cases that are correctly classified at each cutoff. Whereas negative predictive value is the probability of being a true non-case among all the observations that are labeled as a case. Changing the location of the threshold will change the way that observations are classified. As the classifications change, the values of all these performance metrics change as well. So what we see on the plot is the value of the performance metrics at each potential cutoff between 10 and 99%. Let's look at an example starting on the left, the label 10 on the x-axis. So if we set the threshold here, we would end up labeling 10% of the observations with the lowest risk scores as non-cases and all other observations as a case. If we do that, the sensitivity is 95%. We've captured 95% of the true cases. But the PPV is only 67%. Only 2/3 of the observations that we label as a case are truly cases. Negative predictive value is around 58%. We classified 10% of observations as non-cases, but only 58% of those were truly non-cases. This tells us that some true cases have very low risk scores. Specificity is quite low. At this cutoff, we've only captured about 18% of the true non-cases. As we move the threshold higher and higher, PPV and specificity both improve, while sensitivity, the proportion of true cases that are correctly identified, goes down. NPV decreases mildly. Where to set the threshold or how to choose the cut point depends on the practical substantive goal of the work. For example, if we were developing a preliminary screening test, the goal would be to identify all true cases. That is, we would prefer high sensitivity and we'd be less concerned with PPV. To achieve that goal, we would set a probability threshold that was low, perhaps around the 20th percentile of the predicted risk scores. In our drug safety study setting, the FDA likes to see a PPV of 80% or greater. Here is a threshold that yields a PPV of 80%. But you can notice that the PPV is fairly flat in this entire region. Towards the left, it's still a little greater than 78%. Now look at what happens to sensitivity in that region. Sensitivity is the proportion of cases that are correctly identified. If the threshold is up at the 70th percentile of the risk scores, we have a sensitivity of only around 38%. So that means we miss over 60% of the people who truly did experience an anaphylaxis event. So an alternative is to trade off a little bit of PPV in order to greatly increase sensitivity. This would be a regulator decision, not a statistical one. But the takeaway message is that where to draw the threshold depends on what you are trying to accomplish. And cross-validated performance metrics allow you to make an informed decision regarding the trade-offs in choosing any particular cutoff. In this plot, we added the performance metrics for the external KP Northwest validation set, and those are the lines that are labeled with 2s. Overall, the trends are quite similar. You can see that PPV is remarkably consistent across the 2 sites. Specificity is worse at low thresholds, but better at higher thresholds. Sensitivity starts out a little better at KP Northwest, but then falls faster than it does at KP Washington. NPV is similar, but becomes worse at higher classification thresholds. Overall, generalizability of our model to Kaiser Northwest was pretty good. The results are consistent with the expectations set by our 15-fold cross-validated metrics. Differences that we do see are partly what you'd observe when evaluating any model on two different data sets, and partly are a comment on generalizability to a second site. Overall, this work demonstrated the extra predictive power we were able to extract from the EHR text notes using natural language processing. And we also showed that for anaphylaxis, machine learning was better able to capture associations in the data than parametric modeling, and in particular, the Bayesian additive regression tree approach. Now I'd like to stop and acknowledge the many people who contributed to this project at multiple locations throughout the country, including the FDA, Harvard Pilgrim Research Institute, Kaiser Washington, Kaiser Northwest, Vanderbilt University, and Health Corps. We also thank two of the FDA Sentinel data partners, Kaiser Northwest and Kaiser Washington, for permission to use their data. And thank you all for your attention. Now, if there's time, we'd like to open it up to questions and discussion from the audience. Thank you.